Good morning and good afternoon. Thank you for joining the second session of our 2020 virtual user meeting. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentation. By default, all audience members are muted until the end of the presentation, at which time we would open the floor to some questions. It is my pleasure now to introduce Dr. Fernanda Laezza, who will hand over the remaining talk.
All right. Thank you, everybody, and good morning or good afternoon. This is 9 a.m. here in Texas. So in the next 20 minutes, I'd like to give you an overview of what we're going to do with the beautiful PortaPatch 384 that we're going to have soon in loan for the next 9 months. And I'd like also to thank again Nanion for this wonderful opportunity this instrumentation grant that allows users in the academia or industry, but particularly in the academia, to become more familiar with automated planar patch clamp electrophysiology. So I just want to give you first an overview of, okay, something's not. Okay, yeah, an overview of who we are and where we are. So this is the Texas Medical Center, which is located in Houston. I'm a faculty in the Department of Pharmacology at University of Texas Medical Branch, which is part of the TMC. And I don't know if many of you know, but the TMC is one of the largest life sciences cooperative in the world, with over 100,000 employees and 10 million patients per year and 61 research institutions. And to me, it was so really shocking that in the entire TMC, we did not have a automated planar electrophysiology equipment since now. And so I think that the idea of putting together a proposal for the grant presentation, for the grant competition, was really motivated by the need, local need, and obviously individual need of specific investigators. So we're very pleased for this. Okay, something is okay. All right, so so the way we organized our proposal actually is like a multi-user NIH grant for what, I mean, some of you I'm sure are familiar with this format. So we had 6 major users, and I serve as a principal investigator. And this is the list of this is the list of PI involved as major users with different titles. And you can see from the bottom here in the scheme, we have a whole variety of different projects and channels involved that will be studied using the PortaPatch. We also have project involving transporters and even synaptic release complex. So for the rest of the talk, I'm going to focus on my project, which involves development of allosteric modulator for the voltage-gated sodium channel. So now I'm going to be switching to a more specific and more like research talk focused on my project. So, I mean, this is really needless to say to this audience, but I just wanted to reemphasize our overall motivation of pursuing ion channels. So we're really interested in drug discovery, and ion channels definitely are well posed for being target for drug discovery and drug development, and simply because they're so directly involved in transmitting information, especially in the central nervous system and peripheral nervous system, from one cell to another. And also from a translational standpoint, they have a vast majority of ion channels have been directly or indirectly related to human diseases, either as a genetic cause or as a mechanistic link to signaling dysfunction. So they're really essential and important as target for discovery and development. However, if we look at a recent study or statistical analysis of all target that are used for drug development, ion channels don't really make the vast majority. They only are less than 25% as therapeutic targets. So there is still a need, for pursuing ion channel as a therapeutic target and for drug discovery. Now, our lab focuses on voltage-gated sodium channel. This is a beautiful confocal image of a hippocampal neuron, and you can see in red there is a distinct accumulation of fluorescent intensity, which represent the voltage-gated sodium channel. And as you know, these channels are rapidly opening and closing and inactivating in response to membrane depolarization, and they mediate the action potential in both the central nervous system and the peripheral nervous system for the vast majority. Okay, just a second here. Sorry, I'm just moving my, media here. Okay, good. So the interest of our lab has been focusing on searching for different ways of modulating voltage-gated sodium channels. So as you know, the vast majority of drugs or probes that target the voltage-gated sodium channel focus on the pore-forming alpha subunit. So, and that poses a significant number of disadvantages in terms of side effect and lack of specificity. So our research has been focusing on trying to identify new surfaces that could lead to allosteric modulation of the sodium channel, something that has been quite successful for other receptors in the central nervous system. And we have been focusing particularly on protein-protein interaction at the level of the C-terminal tail of the sodium channel. So there are a few different types of protein-protein interactions that occur at the C-terminal tail of the channel, but particularly we've been interested in the intracellular fibrovascular factors, and those are relatively small protein. Even the proportion here doesn't really match with the actual amino acid sequence size. But the IFGF, I'm going to call them like that for the rest of the talk, are directly interacting with the voltage-gated channel C-tail. They regulate not only biophysical property of channels, but also trafficking and cellular targeting. And what is very exciting and important about this interaction is that there are different IFGF, FGF 11, 12, 13, and 14, and most of them, except for 11, that there is no yet clear evidence of that, but the other one, the 12, 13, and 14, they all have been shown to interact with the sodium channel at different isoforms from 1.1, 1.2, 1.3, there are some evidence, 1.5, and 6, 7, and 8. But each interaction really produced a very distinct phenotypic current. So what that told us is that the functional specificity that these interactions are able to produce on sodium channel current are actually likely to occur through a very specific structural interaction. So basically, what motivated us was the the potential structure function specificity for these protein-protein interaction sites. And because we're pharmacology, we thought that this would be a good starting point for potentially developing specific probes and allosteric modulator with unique property of functional modulation of the channel. So among the various different pairs, we focus on FGF14 and NAV1.6 because we knew that FGF14 is highly abundant in the CNS, which has been the focus for our last at least 10 years. Well, I think five to 10 years. But even when I was a postdoc, I was interested in FGF14 and NAV1.6. Importantly, the complex FGF14 and 1.6 channel is highly abundantly expressed in the nucleus accumbens shell and other area of the striatum. And we were particularly interested in this area for therapeutic purposes for developing potential modulator of the sodium channel that could be applicable for depression, light disorder, anxiety, or mood disorder in general. Now, another very important aspect is that we identify through peptide mapping and designing of peptidomimetic hotspot, basically small amino acid residues that are able to mediate the protein-protein interaction. And in doing so, we actually discovered a so-called druggable pocket, which is shown here, filled with this peptidomimetic. And the interaction between FGF14 and sodium channel is functionally relevant. We know that in the nucleus accumbens, if we disrupt the interaction using this peptidomimetic and another molecule we'll talk about later in the talk, we can actually diminish firing of medium spiny neuron in the nucleus accumbens. So again, just to reemphasize, our rationale here was to develop, to explore essentially druggable surfaces at the protein-protein interaction interface between these proteins. We also know that both the sodium channel and the FGF14 are phosphorylated by specific kinases. So really developing probes that could disrupt or potentially increase the interaction or the phosphorylation has been the focus of our research. So here is a pipeline that I put together and thinking about how to move forward in this research, in this drug discovery and drug development in the academic setting. And of course, you know, this is designed with targeting protein-protein interaction as a starting point. But some of these modules, this is really a very modular, obviously, platform. So a lot of these modules can be moved around and substitute whatever is necessary for anybody research. But we came up with a first need, which is developing a very strong and very robust primary screening against the protein-protein interaction interface. And obviously, you know, there are a variety of different chemical library to screen, or even rational design it based on molecular docking or, you know, derivation of peptidomimetic or small molecule from peptide mapping. And then, you know, we have a variety of different counter screening that we do to validate the primary screening assay. And then we have orthogonal screening. And here, what is very important in green, dark green, is the functional assay. So far we have been using manual patch clamp electrophysiology to provide functional validation of any hits identified through the discovery phase. So this is where really automated planar electrophysiology could make a significant impact in accelerating the discovery and development of small molecule. And then obviously, you know, we have classical ex vivo electrophysiology, in vivo studies. And then really what it's shown here in this red pattern area, boxed area, is what we think it could be pursued through small business initiative. But everything from blue to green, it's something that with the help of good equipment and a good team could be developed in the academic setting. And again, this is where the automated Planar patch could provide us an advantage, or can make things happen actually. Here we have I have to touch quite quickly on this. This was really the rationale of the primary screening that we developed. So we were interested in something called the minimal functional domain approach. So it's basically trying to isolate a very small fragment of the sodium channel, particularly the domain that interacts with the FGF14. So we adapted a split luciferase assay, which has been broadly used in cancer research, in order to detect protein-protein interaction using luminescence as a readout. And because the sodium channel is such a large transmembrane protein and it poses difficulty in expressing it, so we express it as in form of a chimera fused with a CD4, which is a single transmembrane protein, and the CTL of the NAV 1.6. So what's great about this assay is that the chimera is really expressed in form of puncta, which really resemble what we can see in vivo in NAV 1.6 in neurons. And the other thing, so this is really reproducing the best we can a physiological environment, even for a reduced primary assay. And something very important for us in terms of drug discovery were the statistical parameters. So we were able, with the help of the high throughput screening core at the Texas Medical Center, to really achieve optimal statistical parameter for an in-cell assay with a Z prime factor of 0.8 and these really great signal to noise, to background, and signal window. And so all of these parameters were really important to make sure that the assay was scalable and could be really seamlessly integrated with other screening platform that we have in the core. So this is the next few slides are just to give you an idea of what we have done so far. Well, we have done other things published, but this is unpublished, all unpublished. So we screen about 50,000 compounds. This is a small molecule, a collection of small molecule library available at the high throughput screening core there. We use the primary screen, the split luciferase assay, as a way of identify hits. And then, as you can see here, this is a summary of the screen, and each dot represent an average of 2 wells. And this was done in 384 well plate with the stably expressing cell lines that we made. We had platforms that were run side by side based on luciferase or fluorescence that are used as counter screening for toxicity and false positive. And then we have a platform that we use on validated hit and powder that is repurchased or resynthesized, and then we do those responses to identify, provisional IC50 or EC50. And then as one of the orthogonal screening that we use is a SPR, surface plasma resonance, which is based on purified protein, so it's much more expensive. It's not really high throughput, but it's very specific and provides a very good quality information on binding parameters. So we're looking at specific KD of these values. We also look at the way in which specific hit are able to disrupt protein-protein interaction using the split luciferase, the SPR. Now, one thing I forgot, it's another orthogonal screening that we do. We counter screen against other IFGF complex with sodium channel, as I told you earlier. There are other interactions such as FGF13 and 1.6 or 1.7, also dimers of respective IFGF. And you can see that it's very impressive, even though these are quite similar structurally, but they're not identical. So we can see a kind of an interesting pattern here of specificity for certain compounds, even within a dimer versus a monomer to sodium channel interface. For instance, for FGF14, you can see this difference here, or this compound, for instance, is active on the dimer but not on the monomer. So what we were postulated in terms of structure function specificity of these protein-protein interaction complex, it really seems to be a valuable hypothesis supported by experimental evidence. And of course, you know, we're interested in all allosteric modulators. So the next step is to do functional validation and using patch clamp electrophysiology. So we have here one example of a compound that we were interested in. We're calling it 7605. This is a small molecule that came out of the screening. And what we did, we used HEC293 cells stably expressing 1.6 and then co-expressing FGF14. And so here, again, we use manual patch. We use manual patch here to look at different parameters of sodium current. We can see, for instance, in particular case, we have a change in tau induced by the compound. We have some changes in voltage dependence of activation and steady state inactivation. We have a very interesting effect on long-term inactivation, which is a phenomenon that in this particular case, when FGF14 is co-expressing 1.6 in HEC cells, is not evident, but the compound is able to induce long-term inactivation. So there is a lot to do. Of course, you know, before I go to the next slide, I just want to mention, obviously, we did ex vivo electrophysiology. This is an important step that we do. We actually do it in an order which is a little different from what I depicted earlier. We do it side by side with the manual patch in HEC293 cells. We want to make sure that we pursue a hit that is functionally relevant in ex vivo slices. Preparation. So here we're looking at the nucleus accumbens slices in medium spiny neuron whole cell patch, and we looked at persistent sodium current that are in these cells are a signature of NAV 1.6. This is not only NAV 1.6, but NAV 1.6 is highly involved in the regulation of these persistent current. So we can see that, as expected, we have a significant decrease in the peak current. The peak sodium transient current. We also obviously look at firing, and we can see there is a significant decrease in maximum firing frequency. And importantly, in the knockout mouse, you know, the FGF14 knockout mouse, so FGF14 obviously is our target, we don't really have any effect of 7605, which really further build confidence in this small molecule as a target of the protein-protein interaction interface that we're pursuing. Now, if we want to move forward, however, you know, really looking at drug development, what we really need to do is really digging into the potency and selectivity against all the other pairs of sodium channel and IFGF to make sure that we can achieve selectivity and obviously enough potency to pursue the compound further and for lead optimization. And this is really where the automated Planar electrophysiology equipment can come into play. And this is really the way we envision we're gonna use the SynchroPatch, and sorry, but we do also have a PortaPatch. And so some of these experiments are currently being tested and optimized using the PortaPatch. But so this is a very simple scheme, a layout of how we're gonna, of our experiment, our experimental plan. So we're gonna have all sodium channel isoform on one row. We're gonna have different IFGF. We're going to match test every combinatorial pair, even though for some pairs like FGF11, there hasn't been any experimental evidence published for interaction, but because there are structurally similar, there is structural similarity between these IFGF, it will be safer to really look at everyone. And then initially we're gonna have one single concentration, and then on selected hits or selected pairs, we're gonna do a 4-point dose response. And what we're gonna do here, we're gonna go back to the result of high throughput screening. We have about 50 compounds that are non-validated, non-toxic validated small molecule that we will pursue and counter screen. And so this has not only a value in the specific campaign against 7605, which we're currently pursuing, but we hope that by doing this counter screening, we identify other potential route of investigation. We're particularly interested in FGF13 because of its role in pain, peripheral pain for that. And we do have peptidomimetics that are targeting FGF13 for pain purposes. But those, these molecules are different, structurally different from the one we have. So basically, to conclude, I just wanted to leave you with this slide and tell you where really automated planar electrophysiology could give us an advantage, at least in the academic setting. I think really here, in the very early phase for discovery screening and for lead optimization, automated planar electrophysiology could make a difference. Not only that, it can allow drug discovery and part of the drug development to be done in the academic setting. This is really different from what was going on, 10 to 15 years ago in the drug discovery field and drug development field where only big pharma were pursuing these aims. So I think is really extremely important that companies like Nanion are able to dialogue with us, with academicians, and really introduce us to your amazing technology. I think that it's going to be a different era in terms of discovery and development, drug discovery and drug development, especially for ion channel with the advent of automated planar electrophysiology, and fortunately and finally its arrival in the academic setting. And this is my acknowledgement. These are people in my lab, present and past members who contributed to the work that I presented today. A great thank to the Institute for Bioscience and Technology at Texas A&M in Houston and the Screening Center. And the other investigator, major user at UT Health, where the SynchroPatch will be located, as well as Rice University. And with that, I'm going to be hopefully answering questions. Thank you.
Thanks, Fernanda, for the interesting conversation and presentation. We're much looking forward to the data that you're going to receive on the SynchroPatch. I now open the floor to a Q&A. For Fernanda, I would just kindly say that perhaps in the chat window, you could write your name if you'd like to ask a question, and then I'd go ahead and unmute yourself, and then you can ask Fernanda the first question.
No questions, I guess.
Um,
Actually, can I ask a question? Maybe that would stimulate some Q&A. Is there anybody in the audience who is currently in the academia and using the SynchroPatch? And if so, can I have a feedback on their experience and their goals?
we had one question that just came in, Fernanda,
Okay, already.
from Mark, and he asks, are there any species differences in the FGF NAV 1.6 hotspots or effects?
Species different. Actually, we have looked only at human for now. So all our construct and our screening are done on human sequences. So we haven't really looked very specifically into other species. That's something extremely important when you use, obviously, construct from different species. Also obviously important when you transfer your discovery into a mouse model. That's definitely important. But yeah, we haven't looked. They're fairly conserved. The intracellular FGF are fairly conserved between rat, mouse, and human. So I would assume that these are actually the hotspot, specifically the hotspot that we pursue are concerned. But that's a good point.
And then maybe I have one open question, which is more on
Yes.
the kind of grant and proposal. Maybe you can speak to a little bit about the grant process for you. Obviously, a lot of research needs to get funded, and maybe you can speak a little bit to the process that you had to go through with the group some time ago in actually submitting this application, and how that kind of deviates from the traditional roadblockers that may exist in pursuing research like what you're looking at pursuing.
So are you talking about the Nanion grant, or are you talking about in general?
Yeah, in general. So looking at the conventional roadway that you would traditionally versus, you know, us as a manufacturer, looking at this as an opportunity.
Oh, okay, I understand. So specifically for the drug development and drug discovery project.
Exactly.
Yeah, so I have to say that I have worked with the Pharma Foundation initially. That was my very first seed grant, and this is really a small amount of money, but it was very instrumental to get started when I started my lab 10 years ago. And then I have been having dialogue, very constructive dialogues, very early on with the NIH. And I've initially worked with NIMH. I mean, some of my funding, so actually at my current funding sources are here. But so I started with NIMH because of our interest in neuropsychiatric disorder and the great need in finding, new target for neuropsychiatric disorder. And NIMH has an exceptional, plan, which includes very specific PAR and RFA to really promote drug discovery in the academic setting. Then for the drug development aspect, which is something I have started to pursue a little more recently, I have founded a small startup company. So I'm pursuing a traditional STTR, SBIR for moving forward in the development. But I have to say again that the NIH, and I can speak also for NIDA, I recently have been awarded a U18 grant, which is exactly doing what I'm describing, taking academician hand by hand and walk with them on helping on developing project that could potentially have a commercialization and a development perspective. So again, the NIH, again, at least NIMH and NIDA have a phenomenal team of individuals and programs that you could pursue.
We had another question that came in, and to paraphrase, you mentioned a little bit during your presentation, a little bit about the PortaPatch, which you have, and the question is asking what various research areas you've used the PortaPatch with.
Okay, so yes, we have it. The problem is that it was ordered a couple of weeks before COVID-19 shut down. And so it was actually delivered much later. So we are just working on it now. So currently what we're doing with the PortaPatch, we are trying to optimize it, especially to really work the health of the cell. The cell conditions is the essential parameter, and because the interface between the cell and the recording platform is identical in the PortaPatch and the SynchroPatch 384. So we're using at the moment the PortaPatch for simply in preparation. But our plan is to use it side by side with the initial functional screening that I've shown you before, so the initial validation of hits. We have been using previously a PortaPatch that a colleague of us has at the Texas Medical Center, just to have an idea how it works, but again, only on the sodium channel so far. Yeah.
Then another question came in. Do you anticipate having a lead compound by the end of the 9 months?
Yes, actually, we already have a lead compound. We are hoping to, well, again, let me step back, okay? Lead compound means a lot of different things in the drug, in the industry versus academia. So we have let's say, a promising lead compound that has micromolar potency. We wanted to optimize it down to nanomolar potency, and we have some evidence of specificity based, and not specificity, selectivity on against other isoforms of sodium channel. And so we anticipate that we will have a better idea. I don't know if we can have a lead compound by the end of the 9th month. It depends when the 9 months starts. We haven't received the SynchroPatch yet, just because we are a multi-user group and we are still trying to, coordinate all the efforts and make sure that when the equipment arrives in Houston, we have everything ready. But so if we start the count by January, we most likely by next summer, which will be a year from now, we'll have most likely we'll have a lead compound. Obviously, it's a matter of resources, right? Because we have a compound, so if we have a good amount of money today, so we can just outsource to any CRO or to drug discovery and drug development core facilities, so we can have compounds synthesized rapidly. And our platform for screening is quite fast, especially up to the functional assay. So that's not a problem. The barrier now is to synthesize the optimized leads.
Then another question from the audience, also kind of touching upon the use of the PortaPatch. Are you having any issues obtaining patches reproducibility on the PortaPatch?
Yeah, so I don't want to say anything negative about the PortaPatch, but I also have to be honest. So right now we're currently fighting for obtaining the quality of cell culture that we need. And I know I don't want to say anything bad, as I mentioned, just because I know people that have used the PortaPatch. I mentioned a colleague of mine Christine Bitton at Baylor College of Management. She has been using very successfully the PortaPatch with many different channels, potassium, calcium, and sodium channel as well. And I know she spent quite a good amount of time in optimizing the culture and worked hand by hand with Nanion. So I am confident that we will achieve our goals. But I have to be honest that really seemed to be the critical issue. There was another issue that we thought was could be a barrier, but it most likely is not going to be a barrier. So we're interested in targeting a cell line that have two transgenes that are co-expressed simultaneously in a stable format. So right now we have sodium channel isoforms. So we have cell lines that express sodium channel isoform, but we don't have a cell line that stably express the intracellular FGF. Actually, we have two. They're on their way. We outsourced them to a CRO, so we're gonna have them soon. So we initially thought to really make all of these combinatorial cell line, but I've recently heard from other investigators with similar needs that using even transient lentivirus transfection is the way to go. So in addition to that, we'll, I'm not sure what happened here, but in addition to pursuing a traditional double stable cell line manufacturing, which will be done for some pairs, we will also use lentivirus. I have no idea what is this. I don't know if you see the same thing. So it's something weird, maybe about my pad. I don't know what it's.
Yeah, sorry about that. I don't know what happened. It's
no idea.
yeah,
Let me see if
someone's on there.
Oh.
Okay. What I'll do then is I'll go ahead and I think that provides a good gateway for us. We'll reproduce this and we'll make this available on demand. And any questions for anyone that may have not attended due to time differences, I'm sure that we'll tag you, Fernanda, along, and then we'll also include that on our website. The next meeting from the Broad Institute will begin at the top of the hour, and we're looking forward to the next talk.
All right, well, thank you so much, and feel free to email me if you have any specific questions.
Thank you so much, Fernanda. Have a
Bye-bye. Thanks.
Take
Bye-bye.
care.